They never had an idea to create a company. Their goal was actually to treat the patients, but the outcomes were so good and a bit of unexpected because they have cold solid tumors that we decided to okay, we need to make this into a company and bring this to more people to treat hundreds and thousands of patients per year. Hello, everyone. This is Lucas, CSO of CuraCell, and you're listening to the Repertoire Room.
Hi, Lucas. Thank you so much for joining me on this podcast on T-cell repertoires. That's the best intro we've had so far in the last 4 episodes. So thank you for that.
That's my pleasure. Thanks for having me.
No worries. Before we dive into who Lucas is, can you shortly tell me a bit about CuraCell?
Yeah, so CuraCell, it's a clinical stage Swedish company. We were founded in 2022, and our main goal is to bring TIL therapy, so tumor infiltrating lymphocyte therapy, to treat cold solid tumors. So that's the goal for the next year.
Okay, an interesting goal for the next year, I feel. Okay, yes. Well, we can have some time later on to really deep dive into what that exactly means. But I also like to start a bit with the person behind the science, behind the companies, etc. So what's your origin story, Lucas? How did you end up actually, or what was your career prior to CuraCell?
Yeah, so I'm originally from Brazil. I'm a pharmacist by training. And then after finishing my studies, I did my master's and PhD working with cell therapy to treat autoimmune diseases, especially type 1 diabetes, multiple sclerosis, and systemic sclerosis. And during my PhD, I started to use TCR technologies to understand how the therapy could rebuild the immune system and make a patient that has an autoimmune repertoire to be rebooted to become more healthy. And during that time, I had the chance to go to France to do an internship. And then I had some fun. It was nice, good publications. Then I decided to do a postdoc abroad. Then I was looking for opportunities, got some positions offered in the US and in Europe. Then I decided to join Sweden to come to Karolinska Institute because it's the homeland of the Nobel Prize. And it's quite a prestigious institution. Then I came to Sweden around 8 years ago. That was not the plan. The plan was to come do a 2 years postdoc and move back to Brazil, but life changes. And then I did my postdoc. Then I changed a bit. Instead of using cells to treat autoimmune diseases, started to use cells to treat cancer. So I worked with gamma delta T cells, with hematopoietic stem cell transplantation for leukemias, and also to work with bispecific T cell engagers. And then after some years of postdoc, working on the academic side, I'm changed and moved to a biotech based here in southern part of Stockholm named Arruda, where they do TCR-Ts to treat also cancer. And then after some years there, I had some fun. I joined CuraCell as their CSO. And for now, in the past 2 years, I've been working here at CuraCell and driving our TIL treatments forward into clinical practice.
Oh, yeah. So that's quite some transitions from Brazil to Sweden. It's already quite a transition, but yeah, also autoimmune and oncology now, although the key point seems to be T cells connecting everything together. Always.
And also TCRs, especially. Yeah,
and TCRs. Yeah, cool. All right. Yeah, you already told me indeed now that you joined CuraCell after it's been founded, but do you have any idea of the origin story there? How did it get started, CuraCell?
Yes, sure, of course. So CuraCell is part of an umbrella. There's a main holder company named Arruda that was founded in 2018. Initially, they had a focus on the mesenchymal stroma cell side, so bringing cell therapy to another indications. But then when our current CEO, Jonas Botterson, joined the company, Arruda, he started to connect different IPs that the company had in the portfolio. And then he brought CuraCell to this umbrella as well. So CuraCell has a bit of an unorthodox development because it co-developed in Sweden and Germany. So there was a lot of exciting research being done in Sweden in a way to expand tumor infiltrating lymphocytes for cold tumors. We're not talking about melanoma or lung cancer. And then because of this connection that they had with Germany by that time, because it was some German researchers here in Sweden, they could put this quite early on into clinical practice. Because in Germany, you are allowed to do what is called, there are many names, named patient treatments or compassionate case use or hospital exemption. So early on, we could not just show that we could create those products, but we could early adopt them into clinical practice. and then see if they work or not, and then tailor-made these products to be even better for cold solid tumors. And that's how the company got started. So IP and ideas and expansion protocols coming from Sweden and early clinical adoption in Germany that made all of this happen, in a very unorthodox way.
Yeah. Well, indeed, my next question was going to be what it's like building a biotech in from Sweden, but I've also learned now that one of the things moving it forward was also the presence in Germany, where you basically were able to very quickly already get into a patient. It's not a clinical trial yet, but very early on in the stage, get some insights on how these kind of therapies would work within a patient, right?
Yeah, exactly. No, honestly, to build up a biotech company in Sweden is quite great because the Karolinska ecosystem is amazing. There's a lot of great scientists around, and cell therapy, it's a big part of it. So there are not only companies with TILs. Actually, 2 out of the 6 clinical stage companies in the world are based in Sweden, which does a NeoGap, which is amazing, but also have TCR companies like Anoka and also CAR-T companies. So there is a lot of R&D, and it's quite easy to build up a team around that because we have a lot of great brains. And cell therapy is basically blossoming in immunotherapy in this ecosystem here in Stockholm area because of Karolinska.
Yeah, so it's Karolinska actually is the glue that connects all of these companies together, but there's just a lot of knowledge in the whole area, it seems.
Yeah, exactly. It's a mix.
Okay. Well, this podcast is about repertoires. You already mentioned the word TCR a couple of times. Can you give us some insights? Are TCR repertoires being used at CuraCell? And if so, how do you use them in daily practice?
Yeah, so we are using quite a lot, to be honest. So TCRs at CuraCell use it in different ways. I would say that we use it quite often for understanding our product and the journey of manufacturing a TIL product. TILs are complicated drugs, and it's complex, not just the clinical application, but also the manufacturing, especially if you want a tailor-made specific products for cold solid tumors. So in cold tumors, you have a few mutations and therefore have a few infiltrating T cells. So we had to adapt and tailor-made our expansion protocol and our platform in order to be useful and applicable to those solid tumors. Therefore, use TCR technology to see how we could manufacture these products. And also when we used and we adopted these products early on, to do the immuno-monitoring of these products inside the patient's body. So because we had this early clinical adoption, we could basically use TCR NGS to sequence the product. And we got many samples collected in the course of the treatment to see the biodistribution, the persistence, and to use some bioinformatic pipelines to see if the TILs are expanding in the tumor microenvironment or not. What is the role of the resident T cells in the TILs that we gave to the patients, and how that is connected with the clinical outcome. So very early on, by using TCR technology, we could already connect some dots about not just the QC of the cells, but also the mechanism of action, showing that these cells can actually infiltrate back to the tumor tissue and drive the observed clinical outcomes. So yeah, we use TCR technologies in a big span.
Okay, so it's both product characterization as well as an immuno-monitoring perspective. And what I'm hearing from your responses, it's this immuno-monitoring is not only something you do on blood samples, on PBMC samples, but you could also get access to tumor samples to basically characterize them.
Yeah, so now we are doing some publications because the clinical data are really, really exciting. So in this cohort of patients that we had an early adoption of our product, we had the patient, for example, with glioblastoma that had a terminal disease. The patient has undergone all possible therapies, but he had a resectable disease. So we could collect a small piece of tumor and we could manufacture a product, which was great because normally you don't find many T cells in a glioblastoma tissue. And that product was very good at killing the cancer cells that could grow from their own patients in a very specific way. And when we infused these TILs to the patient, we could see that after the first infusion, the cells started to decrease in blood because they had a peak of expansion after around 1-2 weeks. And then it started to migrate back to the tumor tissue because the patient responded. The patient had a partial response after the infusion. And that's one of the publications we had this year at Oncology. And then because the patient responded so well to this first therapy, the clinicians decided to induce a complete response by giving a second infusion. And then by giving a second infusion, you could see that we had even higher infiltration of the TIL-derived clonotypes to the patient's tumor. And the patient achieved a complete response. And the same was observed for other indications. So early on, by using the TCR technology, we could learn that the multiple infusion of TILs improves quite substantially the pharmacokinetics profile of the drug. So for hot tumors, you might not need more than one dose. It's basically one shot and done kind of therapy. You have melanoma or lung cancer, which is going to be probably the next approval. You manufacture a product, you get one dose, and you have a good likelihood of success. But for cold tumors, where the tumor microenvironment is very harsh, and you have very few T cells, so it's very hard to manufacture a product. It's something that we use TCR technology to optimize our product development, but also to show that you cannot achieve such good responses with just one dose. You might need more than one dose, most likely 2 or 3, or even combination therapy with checkpoint blockade. So, and that's what we have learned, and now we're publishing some papers. So we have recently published a paper as well with a prostate cancer patient that achieved stable disease after one dose. So we could see the profile of the T-cell derived clonotypes by using TCR technology, expanding and reaching a peak 1 month after infusion, but then decreasing because it's natural of the pharmacokinetics of the cells. But when you hit with a second dose, you get this peak going back again, and the patient achieved a partial response. And then we hit with a third dose, and we combine with checkpoint blockade, because we have learned from other studies that checkpoint blockade is quite synergistic with TILs. And then the patient achieved a complete response, and now for more than 5 years. So the patient came to die, and the patient is alive and kicking. So it was great to be able to use TCR technology, not just to improve the manufacturing for cold solid tumors, but also to have the proof points to show that, yeah, it makes sense to do multiple infusions. And actually, we could show in this manuscript as well how the checkpoint blockade combination not only boosts the TILs that you infuse, but it also brings new players to the game. Because some of the TILs, they might not be so in good shape that could be reinvigorated by the checkpoint blockade, but it's not so bad because they can also bring new players to the game. So you create a more strong anti-tumor microenvironment. So we are using TCR technology constantly, not just for the manufacturing QC, but also to understand the mechanism of action of our drug.
Yeah, all of this sounds extremely exciting to get this kind of data, even from N=1 patients. Like you're very, very. It feels almost like you're bootstrapping things and figuring out, like, oh yeah, this is the response. What can we do next? Of course, in collaboration with the physician. It feels that you're learning a lot with these single patients.
I mean, that was magnificent. So the physicians team based in Germany, they were quite keen to help patients. That's their main goal. They never had an idea to create a company. Their goal was actually to treat the patients, but the outcomes were so good and a bit of unexpected because they have cold solid tumors. They decided to, like, okay, we need to make this into a company and bring this to more people to treat hundreds and thousands of patients per year. So early on, they realized that we need to collect blood samples across the course of the treatment. If possible, get some biopsies here and there. Then we could learn why this patient responded and why this patient not responded, and if it makes sense to make a TIL product for cold tumors. And we learned that, yes, it makes sense.
Yeah, that's extremely exciting. Yeah. It seems that, you are applying T-cell technologies or TCR sequencing quite a lot then. At ImmuneWatch, I always look at these things from maybe an antigen specificity angle, but it seems that maybe one of the key outcomes or measures that you are looking for is like, okay, what TCRs do we have in our TIL products? And do they migrate to the tumor samples, to the TIL environment, and do we also see them circulating? Is that correct? Is that one of the main outcomes you're looking for?
Yeah, it is. Of course, we are quite keen to know about the specificity of those guys, because TILs work because you can actually fish tumor-specific T cells from that tumor specimen. You can expand ex vivo, and they do the job of controlling or clearing the tumor. But it's really hard, and the biotechnological tools, of course, ImmunoArt's trying to crack that down, and it is great, but it's really hard. And our main goal with these technologies used in the past is to actually connect the dots to make sure that we can manufacture a good product, not focusing on the reactivity itself to specific antigens, but to show that those cells can actually kill autologous tumor cell lines for all those patients who could actually grow some tumor cells, and show that the products that we made is actually tumor specific, and have all the controls to show that it's only specific in killing the tumor cells, not healthy cells. But also to understand why it's so hard to treat cold tumors is because of the tumor microenvironment. So you need to hit the tumors not just with one infusion, but multiple. If you think about it, when you do chemotherapy, you have many cycles. If you do checkpoint therapy, you do many cycles. Why TIL therapy and T-cell therapy with just one shot? Of course, it is like that because the manufacturing is really complicated, so it's scalable. But what we have a very simple manufacturing process. So all the complexity on the manufacturing, so instead of engineering cells, instead of selecting tube types, we have an expression protocol that does that by itself. So then we can have a bit of more complexity in the clinical application, as long as it works, and it does work. So the goal of the clinical trial that we're going to run next year is to actually bring all this experience we had with the patient-named cohort. That is not a clinical trial. Everything was adapted for each patient, to put that into clinical practice and see if you can see the same great outcomes.
Yeah. Okay. Tell me a bit more about that, because that's apparently the big next thing in the coming years for CuraCell. The going into a real clinical trial there. What's the goal there? Is there some kind of target disease that you're looking at? Can you tell me a bit more?
Yeah, that's a really good question. So after all this knowledge we have built over the years in Sweden and Germany, we decided to go through the regular track for getting a drug approved, which is basically to get a manufacturing license for our CDMO, because during this time of co-development, we also started the CDMO business. We had a partnership in Germany to manufacture these drugs. So all the knowledge from the expression protocols in Sweden were transferred to the CDMO, and that they could manufacture this drug there. And there they could manufacture for the patient named. So now we are following the regular track to run a clinical trial. So based on our experience and also the market, we decided to go after prostate cancer. There is no immunotherapy approved for prostate because it's a very cold tumor. Checkpoint blockade doesn't work, and we had a very good experience with just one single patient, but the experience was magnificent. And also for colorectal cancer, especially for microsatellite stable colorectal cancer, where also immunotherapies are not available and it's very hard to manufacture product. So we did all the validation runs, manufacturing licenses granted, the clinical trial is approved. And we have a very unorthodox clinical trial as well, not just the development, which is about the multiple dosing of TILs. What we have learned in our patient-named cohort, despite not being a clinical trial by itself, is that you most likely not going to get good clinical responses by doing just one infusion. So we are going to do multiple infusions, and if necessary, combine with checkpoint blockade. So the goal is to actually drive good responses for long term, as we have seen for some of the patients. So that's the goal for next year.
Okay, sounds like an amazing goal. Will this be run in Germany then, or is it something worldwide? Or
good question. All the R&D is done in Sweden. We keep running new optimizations in our product, in our protocol, but all the manufacturing and clinical application is done in Germany, especially in Frankfurt.
Right, Frankfurt. Okay, interesting. Will T-cell sequencing then also be a part of like the evaluation, because T-cell sequencing is usually presented as a research-only tool. It also has to go to the regulatory checks to get it really be approved in clinical trial, but it feels like it has been giving you a lot of value in figuring out whether it is indeed working next to just a tumor shrinkage, I guess. Will it still be part of the clinical trial protocols?
Yes, that's a good point. So because I am the CSO of the company and I have a lot of love for TCR sequencing, I made sure that in the exploratory endpoints of our clinical trial, we're going to run TCR sequencing all the time to make sure that, first of all, we have multiple dosing to show that the product is the same. Imagine if the TCR composition is different among the doses, then we have different products. But of course, in the validation runs and all the preclinical development, we have products that are 99.9% equal in TCR composition, phenotype, characteristics. It looks great. So that's one application we're going to have in the clinical trial to show to the regulatory agencies that we are doing multiple dosing of the same drug and not having crazy TCRs and oligoclonal expansions in different doses. No, it's exactly the same. And the second one is to show that how good it is to do the multiple dosing and the synergistic effect of the multiple dosing plus checkpoint blockade. So I would say that TCR sequencing is going to be one of the pillars of the execution of this clinical trial. Of course, the goal is to show the safety of the multiple dosing. We know it's safe because we have done this before in many patients in different indications. And patients more frail than actually the ones we're going to include in the clinical trial, because all those patients that had terminal disease for real. And yeah, basically to track the cells and show that it is a good approach. And hopefully the patients will have a good response. The focus is safety. We hope to see some efficacy. And we're going to use TCR technology to put everything together.
Well, that makes me super happy, because I've been talking to a lot of people who are starting to use T-cell sequencing. It feels like it's a methodology that's rising in popularity, and it feels that you are at the forefront of really using that to, of course, main goal is safety, as you mentioned, but also to get some hints of efficacy. Although I also like the first part where you mentioned that you're going to use it as a way to show that the product is exactly the same. How does that actually work? Because you basically have multiple infusions. These are different time points. Do you have to create the product every time again? Is it that how do you say this? Is it that repeatable, the process, to get always exactly the same product?
Yeah, that's the cool thing about our technology. So we have an expansion platform, which is named Cytoply. That is an engineered process in a way that from one single piece of tumor, can be either a biopsy or a surgical specimen, we can manufacture many, many doses that are exactly the same. So it was already complicated to manufacture one dose for a cold solid tumor, but now we crack down all this complexity into a very simple but not simplistic way of expanding the cells, that we could generate multiple infusions. In the patient-named cohort, we have infused more than 9 times into a patient to induce a really long-term clinical response. It was extremely safe, efficacious, because the patient was about to die and I was alive and kicking. And we could use the TCR technology to show that all the doses were exactly the same. And it's something that we always want to show to the regulatory agencies, because in our field, the process is a product. So we are making sure that the Cytoply platform, it's quite reliable, it's simple, it's scalable, it's great, and it's tailor-made for cold tumors, and it gives you always the same dose as long as we use the same tumor specimen to manufacture.
Okay. That's also great to look into the future, I guess, then, that you could apply it now here in Germany, but maybe in the future it can be ported to other places and get exactly the same outcomes from the same starting material, right?
Exactly, yeah.
Okay, so 2026 is going to be an exciting year for CuraCell. It's going to be the launch of the clinical trial, but when do you have an estimate on when they will be concluded then?
By the end of the year, most likely, yeah. It's going to be prostate and colorectal cancer, like I said. It's going to be 12 to 16 patients in total. The beginning is going to be more staggering approach to show, instead of doing a dose escalation like normally other companies do, especially on the TCR-T and CAR-T area. The regulatory agency in Germany said that that's okay, we can do full dosing from the beginning, but it's staggering. So we treat the first patient fully, show the safety, do the second and third. So the beginning is going to be a bit more staggered and taking a long pace. But after that, we're going to speed up and treat many patients in a parallel way. So by the end of the year, we're expected to have a lot of patients treated.
Okay, so people have to definitely watch out for you guys at conferences like ESMO or AACR, figuring out what the responses are towards these therapies.
Yeah, so far we have been showing those conferences our patient-named treatments, showing not just TCR sequencing, but exome sequencing and RNA-seq, showing all the possible mechanisms of action, and the publications are coming slowly. Like I said, prostate is published, glioblastoma, and we have a pancreatic cancer patient as well treated with great outcomes that is coming out soon. And now that is completed, now the idea is to look to the future and show the clinical, hopefully good clinical outcomes in this clinical trial, and use the TCR technology to understand how that was done.
This was super insightful. I really like to learn how people are applying these kind of technologies to improve their own technology. And here it has a direct effect in your named trials. It feels like you've been able to use this technology to figure out what's next and really help save these patients' lives. So this has been a super insightful conversation. Thank you for that, Lucas. Where can people find out more about CuraCell or you if they want to connect?
And that's my pleasure. Thanks for having me. You can find us on LinkedIn or in our website, curacell.se.
Okay.
Yeah.
Interesting. Swedish, of course.
I'm based in Sweden.
Exactly. I always like to wrap up this podcast with maybe some kind of, I think it's a fun question. I don't know whether you like it, but what would you be doing if you were not working as a CSO in a biotech startup?
Oh, yeah, that's a good one. I like academia as well. I love being in biotech because it's more applicable, but I'm happy being an academic. But if I was not in academia, I love music. Maybe I'm a bad musician, but I have been playing music my entire life. Maybe be a musician, that would be fun. Yeah, I have played in many bands, many styles, so why not to be a musician?
Okay, that also sounds like an exciting career. But I'm glad you chose for this career because I feel like we can expect some interesting more stuff coming out of CuraCell in the coming years. Thanks for joining me, Lucas, and I'll definitely see you around.
Thanks a lot, Sander. Big pleasure.